Great. So thanks everybody for coming out tonight. Very excited for the program that we have. Maybe say first off, big thanks to Flagship for hosting the evening tonight and providing the food and drinks. Jacqueline, would you like to say a couple words?
I can just say a few quick words here. My name is Jacqueline Dunphy. I'm a senior associate at Flagship. Been there for 5 years now, and we're just really happy to be a part of this community. So thank you so much for having us. I just want to let you know that we have many companies that are hiring in the protein design space. You can feel free to check out our website for more details. And also over the summer, we have a summer fellowship. Anybody who's working on their PhD or an MD or postdoc is eligible to apply for that. And really, it's a 3-month-long experience learning how we do company building and entrepreneurship. So feel free to find me afterward and we can talk about it more. Thank you. And thanks to Chris for allowing us to sponsor tonight. So thank you.
Awesome. All right, so our speaker tonight, I'm really thrilled that Bruno Correia is in Boston this year for a sabbatical. So for those of you who don't know, Bruno is a group leader right now, EPFL, Lausanne, Switzerland. And Bruno, I would say, is really at the cutting edge of protein design methods development, at the forefront of bringing machine learning methods into protein design space and making those accessible to other folks. I'm sure Bruno is turning bright red by saying nice things about him. He's going to come cut me off. So I'll get out of the way. Bruno,
thanks a lot.
Excited for your talk.
It's always very dangerous to be in the cutting edge because you can cut yourself, right? So sort of like this thing. All right. So yeah, we actually had put. We talked about how, proficient computational biologists are with their own computers, right? Which is not much. All right, we had the thank you slide for Jacqueline for giving us foods and drinks. Okay, and this is good. So Jacqueline, thank you. And so now let's get to kind of business. So thanks a lot, everybody, for being here tonight. Thanks a lot for all the people that are also online. And thank you, Chris and Nick and Sergey, for a lot of fun times in Boston, and also for organizing this initiative and for letting somebody that it's a little bit of an outsider that doesn't really live in Boston to sort of like present this stuff here. And yeah, so as Chris mentioned, in fact, this year I've been kind of local in Boston, if you will, where I've been spending my sabbatical year at Radcliffe Institute in Harvard. And so today I'll tell you, but actually most of the action or a lot of the action is still happening in Switzerland. And so today I'll tell you about the efforts and some of the problems that we have worked through the years ever since I started my lab. And I like to think about the common denominator of my talks on how do we actually think about new ways of representing proteins, and how can we actually use to understand those new representations for understanding what function do these proteins actually have, and how can we actually design new function, right? So today I'll tell you about a few things, or at least I will try to tell you about a few things. If we sort of like run out of time, that's okay. But I'll try to cover a few aspects on how can we actually design proteins for very applied purposes in biomedical problems, and then we'll go more sort of like to the very basic and fundamental aspects that we're trying to understand by using novel methods for the novel design of protein structures, and then also in terms of using surface fingerprints for protein prediction and design. And if I have the time, I'll tell you a bit more about how we can actually use these things for ligand discovery. So for this kind of crowd, or at least for those of you that try to design proteins on a daily basis, this slide comes with no news, but yet it's important to remind that the problem that we face here is daunting, right? Clearly, if you want to start by thinking about the many possibilities that are available for just designing a protein of 100 amino acids, you very quickly run into a number of possibilities that is many orders of magnitude much higher than what is estimated number of atoms in the universe. Now, something that has been an interesting realization as we keep enriching our databases of protein sequences is that, in fact, nature has not necessarily sampled a lot of this space very extensively. And really what we find is mostly islands. If you want to think about the protein space as sort of like this large volume of wide possibilities, what we really find in natural organisms is proteins that are in a way related to one another, and very or more rarely we can actually get out of those islands and for instance sample a new point here. And so I'll tell you a lot about how we're trying to do this during this talk. Now, again, for those of you that are not protein designers, essentially in a very approximated and simplistic perspective of things, the protein design exercise, if you're using structures to guide your design, looks like this, right? And again, the exercise here is how can we find a particular sequence which adopts a particular structure, and hopefully that particular structure at some point will have some type of function. Now, maybe something that we are not, sometimes so interested about, but still one of the main motivations of the protein design exercise, at least in my mind, remains as a test to how well we understand the principles of how proteins fold and work. And despite all the great advances that the field has seen, I'm not necessarily sure that we actually understand better how proteins work. So this is sort of like something that keeps me awake at night very often. Now, clearly, even if we don't understand very well how they work, oftentimes we can still design proteins for a lot of biotechnological applications and a lot of bio-related applications, and one thing doesn't really impair the other. Now, just to give you an idea, of course, that here, just showing you perhaps a little bit the landscape on how different types of activities that proteins can have. And this is really what is fascinating about these molecules, is that, in a way, they can do pretty much everything and anything from communication between cells to chemistry to structure to defense. So really, it is a very interesting endeavor to try to engineer novel function into these novel design proteins. So I'll go straight to one of the first applications that we worked a few years back in the lab, which was to try to design components to control CAR T cell activities, and then I'll go from there more to the methodological aspects. And so here, many of you, I'm sure, have heard about what CAR T cells are. Ultimately, they're essentially, I would call them almost like a miracle of biotechnology, where you can take cells out of people, engineer them out of body, and then reinfuse them into patients, and they go and do their job. And really, this is just appreciating how far we're getting. This is really quite remarkable. Now, what these cells are engineered with is basically with some type of receptor that then endows them with the capability to go and chase tumor cells and eliminate them. Now, of course, with a lot of possibilities come also a lot of risks, and in some of these types of engineered cells, a lot of safety concerns arise, because clearly once you put some cells back into people's bodies, you really don't know what's going to happen. And here, basically, what we thought about, or in collaboration with other people that were close to us, it would really be of interest to try to engineer components which would allow us to exogenally control the activity of these engineered T cells. And so, what we did here, and just to give you a framework on how we went about this problem, what we create was what we call the stop CAR. And again, without going too much into the details of what this is, but to give you the high-level explanation. So a chimeric antigen receptor oftentimes is composed by a recognition domain, which is responsible by identifying the tumor antigen at the surface of tumor cells, and then a number of linkers and also a few signaling domains. And so what we did here was to adopt a split architecture, which other people have come up with. But our idea here was to design components which could be responsible to a small molecule. And once we have this small molecule present, and this will force a physical separation between these two polypeptides in a way that the recognition domain is no longer physically connected to the signaling domain, and this would stop the signaling activity of these T cells. And so in the way that we did this, we used a lot of the structural information that was around, and we used this BCL-XL class of proteins where we knew very well structurally where the drug was binding, And also we had information about which kind of motifs could actually engage the same site that was being engaged by a number of ligands, one of them being a clinically approved drug. And essentially what we did was to basically to take this motif, input it into a folded domain and create a binder out of it. And I will not show you too much of the structural characterization or the biochemical characterization, but eventually this ended up being a picomolar binder. And the structure that we predicted or the complex that we designed was in fact correct. And so jumping very quickly into how do we actually test if these components do what they are supposed to do. Here, the idea was, well, okay, so if we have cells that kill in the absence of the drug, when we put the drug into these cells, we should be able to stop the killing activity of these cells. And that's essentially what you're seeing in this plot, where we're quantifying how much these cells are able to kill in the absence of the drug and in the presence of the drug. And you see that, in fact, we see the killing activity coming down, ultimately validating that this design mechanism was right. Now, of course, this was just cell-based assays. Another type of assays which becomes even more interesting is when we actually infuse these cells into mouse models for solid tumors. And here, this readout is a bit more convoluted, but ultimately, what you see is that the stop CAR, in the absence of the drug, is able to control the growth of the tumor on its own. But actually, if you give the drug, now the tumor escapes and starts growing, meaning again that you are able to control the activity of these T cells in vivo. Another experiment to show you a bit the periodicity or the responsiveness of these T cells. If you give the drug and you withdraw, and if you see things responding, just basically showing reversibility. Now, so that's basically where the story showed up. But then you start sort of like thinking, well, what else can we actually do with these components? And that's when we started sort of like thinking, well, perhaps one of the very interesting classes of drugs that are around are biologics. But if you think about biologics, you know, they're starting to get pretty sophisticated. A lot of people can do many different things with biologics, but what we thought about here is to do basically a biologic where we could use a small molecule to basically remove it from circulation. And let's say that a biological is having some kind of toxic activity, and you just want to get rid of it. And in this case, what we created was this two-part biologic where you have this chemically responsive modules, which are again are the same that we use in the T cells, and then ultimately this would disrupt this binding. So, and basically by disrupting this this complex, what you get rid of is for instance things like effector functions, half-lives, and avidities, and basically getting rid of this drug much faster than what you would in case you wouldn't have this switch into it. And then eventually we tried to validate that this constructs that look like they still have the same biological activity. So here you see a T cell assay in terms of proliferation. So if you use IL-15 or what we call this SWIL, which basically is just an IL-15 mounted in an FC backbone, they seem to have exactly the same activity. And then ultimately we wanted to go in vivo and try to see, does the drug in fact clear the antibody that we engineered? And basically that's the results that you see here, that if you give the drug if you give the antibody to the mice and also the drug, ultimately you are able to clear up the doses much faster than if the drug just by itself. Okay, so I'll just close very quickly this part and then move to a different part of the talk. And the idea here was just that, you know, by using components that are already available to us, we can in fact repurpose a lot of activities in native proteins, even without going into exercises of de novo design and fold exploration. It is possible to, in fact, encode a lot of these different functionalities into proteins. But clearly here, you know, we only tried one CAR architecture, and there's still some limitations on what we're doing. So, for instance, we realized that the response to the drug of this CAR T cells was in fact very slow. So, you know, sometimes if you really want to stop something, you want to give the drug and you want to see kind of like an immediate response. And there's many reasons why this slowness can actually be happening. But we have in mind some other different types of architectures, which basically could potentially allow us to stop the response of these T cells in a much faster way. And of course, you know, there's always this conversation that when I tell to people that, well, you know, look, we designed this off CAR, nobody really gets too excited about it. It's more like, oh, but this turns something off. But really what we would love to do is to turn things on. well, and we've done some preliminary advances, but we're not quite there. So that's to leave you maybe for, news for some other time. Okay, so but now thinking again about, please go ahead. Mhm. Good question. And it does act in another protein. In fact, it's an anti-cancer drug, anti-lymphoma drug, and it basically targets BCL2. It depends on the variant of the drug that it's, and basically the reason why it targets BCL2 is because BCL2 is sort of like an anti-apoptotic or an apoptotic protein, and basically when this drug goes there, it allows the cancer cells to undergo apoptosis. So now it depends. Some cells are very dependent on BCL2 activity to survive. T cells don't seem to care so much about venetoclax, in this case is the name of the drug, what it does to them. So, and then it actually varies, right? If you test this drug in primary T cells, they don't care at all. They just sort of like proliferate. If you test them in a cell line, completely kills them. So there are a few things, and of course this is an issue. But as far as we could tell, in terms of sort of like the basic controls of drug only, up to certain concentrations, you were fine. Of course, above certain concentrations, this drug is also not, it's not aspirin. It does become toxic, right? So there is some limitations over there. Okay, so let me tell you a bit about some of these ideas in terms of de novo computational design and the work that we've been doing. And clearly casting the problem, and as I already told you a few slides back, so nature provides us a lot of glimpse into natural sequences. Many of these glimpses or many of the sequences seem to cluster in these islands, and clearly the only reason why we want to do de novo protein design is to explore uncharted space, right? To see exactly what is there, right? And clearly in the last years, and now this slide, I think it's beyond old, but you can imagine that there are a lot of de novo designs and a lot of de novo proteins that have been designed, and this has obviously been a success. Now, one thing that remains true is that clearly we are able to very structurally accurately design these proteins, but many of them are still non-functional, although this also is becoming less true. But it not only tells us something about the methods that we're using, but also how well do we understand how to encode function into proteins. And I would probably argue right now that not very well. But nevertheless, I will tell you a few of the aspects that we try to develop in order to encode function into this protein. So, and one of our first efforts into this was what we call this topo builder. And basically the topo builder right now seems ancient, although it was only a few years back. But the topo builder was basically this computer program or this protocol where it would represent proteins at different levels. And the levels that we're talking about here is you would have a 2D topology definition of the protein that you would like to design. And you would just define this using basically a very topological, simple scheme about how should this protein look like. Now, one detail that you should have here, we always thought about embedding motifs into these proteins. And so you see here that we have a motif, which in this case is an epitope recognized by an antibody. We won't go there too much today, but you see how this is now embedded. So this is the native motif. Now, it's not sufficient for you just to know which kind of secondary structure elements you would like to build into your protein, but you also need to know to connect them. And there's a number of ways that you can do this. So this topo builder idea would give you the capability of connecting different secondary structure elements as you in an extensive manner. Once you have this information, then you could actually go into a 3D space and translate all this information into what we like to call a protein sketch. You'll hear a lot about sketches in the next slides. And then once you have this sketch, then you could do something that would look more or less like this. And I already kind of explained you what it is, but here you're going to see a bit better in this movie, hopefully, where we take basically one of these motifs, which is highlighted there in blue. It is recognized by an antibody that has a number of different advantages. And basically you start building a protein as you go, just by sort of like defining which sketch of this protein you want to look at. So once you have the sketch, then you sample different folding configurations, and once you converge into a folded configuration, then you try to optimize the sequence. So this is basically our first approach into this problem, and we've done a bunch of things with this. Particularly worked from the two first graduate students in the lab, where took this method and took a bunch of what we can call, broadly speaking, functional motifs from proteins embedded into different protein scaffolds, and then we tested their activity in order to see how they can actually elicit different types of antibodies that could then neutralize a virus. But this is probably a story for a different day, or maybe a story that we don't want to talk too much about. Now. What was one of the limitations of what we were doing by then? So as I told you, the sketches here play a huge role. So, and the sketches were basically what was defining what would the proteins look like. So what we started realizing very early is that our sketches were garbage. And as you know, when you pass garbage into a computer, what comes out is not much better than garbage. And so we saw here that, you know, these proteins weren't really native proteins. And I'll get back to this. So what we did here is was basically to try to embed features into these backbones, which would make these backbones look more native-like backbones. And once we had then this native like backbones, then we could actually now fold them and use them much more efficiently to search for sequences which could result in folded proteins. Now, this was good, and you know, and we used a lot of you know, one tool that was critical here, which is called master, and it's good that Bill is here with us today, because there's a lot of things that relate to Bill's research. And I think a lot of concepts that he was the one pioneering, which we learned a lot about in order to drive our own research. And so here we used master in order to basically, of course, this was not just Bill, it was Bill and Gevorg, exactly, right? And it was not just Bill. Bill is just a familiar face, but Gevorg probably did the work. So, and so that's what we used in order to actually get sort of like this this naturalization of the motifs, right? So, but now, you know, this was a pain to run, I can tell you, right? You do not want to install this in your cluster and sort of like make these things work. So you know, we started sort of like thinking, well, there's probably other things we could do, and this is when also work from another familiar face that is here in the audience, which is Sergey, which participated a lot in the development of this TR Rosetta, and we started seeing what was happening here, right? So like, so clearly what was happening here is that now we can actually use and guide this neural networks to basically build us backbones depending on contact maps. And this then takes us to what we called the third iteration of what I'm going to tell you about. So a long story. But essentially what we realized with our naive sketches, and I'm going to spend a lot of time in this slide, I will just tell you so. It was that they actually didn't look like native proteins. And this brings us to concepts of designability. And that's where Bill comes in, right? So Bill has been sort of like working a lot on concepts of designability, right? And I feel like here, this is perhaps, at least in our lab, this has been the poster child for for a template that has the lack of designability. So you see that the patterns, the structural patterns, both in terms of distances as well as in terms of angles from the side chains are all messed up. So if you compare this type of maps with a map of a real protein, although this sketch was actually derived from the real protein, you pretty much lost all the information that you needed to design the protein. So our question here is, how can we actually train a network to start from a blurry map like this and go to a map that looks like that, right? And our answer to this was basically something that also now feels very old. But we basically implemented this network that we call Genesis, which was a variational autoencoder, which basically was trained to do this. Start with something that is non-designable and guide it until the designable space. And that's what we did here. And so, and let me try to convince you that this actually works. Although, it's not sort of like, it's not the solution for all the problems we have, but in particular situations, it might be very helpful. So the way that this backbone construction looks right now is you pipe in a sketch, which clearly misses all the proteins of designable backbones. You pass it into Genesis. Now you have a better map, and then this map can be piped into TR Rosetta, and then you basically get a structure out. And so we of course did some tests using different types of folds, and so I will just basically draw your attention to this plots right here, in particular to the test sets, which are basically folds that this network has not seen, and they're basically fractionated in three types of folds: helical, strands only, and helical and strand. And also then there's a few things that you should see here in three different colors, where one, you go sketch, Genesis, design, sketch, TR Rosetta, design, or basically a combination of thereof. And what you see in this, at least in these two plots, is that when you only use either Genesis or the TR Rosetta, starting from the sketch, you never get as good backbones or designs even as when you do a combination of their both. Now, it is also important to mention that in this case, we're not doing so great. So things are actually kind of breaking down a little bit when we're doing mixed helical and beta strands. But now, maybe you're thinking, well, why do you really want to spend your time doing this if perhaps there are so many other algorithms right now that can do so many things? Well, the reason why we wanted to do this was to basically have a handle on the fold space that we can actually sample. And so we wanted to use this tool to basically force it to create folds which either don't exist or are very rare in nature. Please. Yes. No, there was no reason. In fact, I think we only did it because you did it. So we just did whatever you did. That was basically, yeah, we just took whatever TR Rosetta was handling better, what seemed to be more informative. So in this case, we didn't really innovate so much, honestly. Oh, you mean 0. So what is the TM score that we're comparing to, right? Exactly. So let's see. So here, what we are comparing to is basically, in this case, you start with a sketch, and then you compare to the native fold itself. Okay. And so in this already, whenever you have sketches, your TM scores actually get pretty distorted already to start with. Yeah, exactly. Exactly. So that's how you do it. Okay, so again, we here also learned from work from other people, and particularly Willie Taylor, that had this paper many years ago about the dark matter of the protein fold space, and we got in touch with Willie. He went into his hard drives and sort of like dig up some dark folds, as he likes to call them, for us, and we're sort of like, okay, well, let's try to see if we can actually design these dark folds. And now, the conversation about novel folds, it's always a complex conversation, because for some people, what is a novel fold, for other is just an old fold. So, of course, here what we did was basically, well, in this case, we used the metrics of RMST, which is also not the most optimistic metric or not the most accurate metrics, but we have also done the same type of exercise with TM assessments. And essentially what I can tell you is that many of these folds, in fact, both in terms of RMSD, if you compare it to anything that is present on the PVB or in the AlphaFold database, the matches that you find, structurally speaking, are not similar at all. And the same kind of result comes from a TM score assessment. Okay, so we're ready to do this with Genesis. We have a sketch. We can make it native. We pipe those maps into TR Rosetta, and then we generate sequences. And again, we ask AlphaFold, how many of these folds, in fact, seem to fold back and with high confidence. And that's basically the plots that you're seeing here. So as you see, the sequences that we generate, they do seem to be what they are predicted back very accurately by AlphaFold with high confidences for both of the 3 dark folds that we're trying. And then we go ahead and we actually, again, so sort of like a very heavy loaded slide, but we tried 32 of these designs. And in fact, what was interesting to see is that there was 19 promising candidates in 32 designs. And again, promising is a very broad term because clearly if you look at this guy, the size exclusion chromatography. So for those of you that are not super familiar with this type of data, what you're seeing here is size exclusion chromatography, CD spectroscopy to see if the protein is folded, and thermal stability. So, and this is what you're seeing for these different dark folds. Clearly here, this is not a really great protein. This is also not a really great protein. So there's a mix of everything. But in this mix, you can also find very nicely behaved proteins like the one that you see here. Please. Yes, absolutely. And so basically, and I think this is kind of interesting, right? Because I actually think that, and that's a very interesting question. I actually think that what made the difference here was the fact that the backbones were actually designable, and that helped the sequence search. And so I don't think we did sort of like, a huge miracle. We just basically provide the best starting points we could, and that just helped the sequence search. Okay, so let's see. So okay, that's probably not the closest baseline that one can do, but let's say that you provide a sketch, which now doesn't have any native features. Typically, the sequences that you find that fit those sketch, they are not very well predicted by AlphaFold. So we haven't tested many of those experimentally, because you never test what you don't think it looks good, which is always a little bit of an issue, by the way. But generally, you don't get so much of a success rate in terms of the back prediction in the computer. Okay. Okay, very good. So let's see. So okay, so I have like 5 minutes, right? All right, so let me just wrap up this part. And so the idea here is that one of the things that I like about this pipeline is that in a way, it is very modular. So you actually kind of understand which part of the pipeline is doing what. And so you can actually see where you're gaining attributes into the proteins that you're designing or losing them. And in this way, I really feel like perhaps one common denominator, not to this approach, but many of the approaches that we see these days, being those based in AlphaFold or Rosetta Fold, I think like the really great lift that we did is not really, oh, this is machine learning or whatever. No, it's just that these algorithms, they actually can make backbones that look like real proteins. And that really helps the sequence search. And I think that's part of the reason why you also see success rates increasing. And I think in general, I don't think people oftentimes speak too much about that. Let me just sort of like mention it that. Okay, so let me take you to the last part of the talk, and I'll try to be not super extensive, but it's again about some of the exercises that we've been doing in protein representations, and now we've been using this to engineer different properties in proteins. So for many of you, which I'm sure you work with proteins, you're all familiar with how we can actually represent protein structures and the type of different levels of resolution that we can have. And the next work that I'm going to show you is basically all the efforts that we've been doing in order to try to learn features in protein surfaces that can help us to better predict what these proteins can do, and particularly in functions that involve molecular recognition. So everything that is related to protein-protein or protein-ligand interaction, but mostly protein-protein interaction. And so, and the analogy that I like to do is essentially this, right? So now if we have sort of like all these powerful algorithms that can learn these patterns from pictures, right? In this case, for instance, in faces, and already you can learn if a person is happy, where does it come from, all these kinds of things. Could we actually do the same thing in proteins? And essentially the way that we realized this in the computer was in collaboration with Michael Bronstein. And what we created was this molecular surface interaction fingerprint, which we called massive. And again, these fingerprints should not be very confusing to you. All that they are is basically a bunch of numerical vectors which summarize information about protein surfaces. And so, what we do with this algorithm is basically to represent the solvent excluded surface of protein structures. We segment these surfaces into radial patches, as you see here represented, and then we enrich them with features. And the features that we enrich each of the points of these patches is basically geometric features. So basically types of measurements which tell you if a protein is concave, convex, plane, and also together with the chemical feature. So as we all know, sort of like in our intuition, we all basically have this built-in understanding that for proteins to interact with one another, they have to be compatible. And compatibility oftentimes is about shape compatibility, but also chemical compatibility. So it's not one or the other. It has to be really a conjunction of both. Now, then all this information then can be piped to a type of neural network which comes from the geometric deep learning and computer vision fields, where then once you have this data, you can actually compact it into fingerprint descriptors, which are those vectors that I was telling you about. And once you have those vectors, then you can actually train them for different types of predictions. So in this case, you can either try to predict what is the specificity of a ligand, of a pocket for different ligands, what types of sites in a protein structure are more bindable, so they're more likely to bind other proteins, and also you can do docking. So I won't tell you too much about this. This has been published also now a few years back, but I will only tell you one thing. We were not that much better than other algorithms. In fact, in many of them, we weren't better, but we were just faster. So this data structure allowed us to be very fast, and this will come into play in the next thing that I will tell you. So ultimately, the aim, everything that we did until now in terms of prediction was basically just a way to try to get to design. And so what I will tell you next is our efforts in order to design the novel protein-protein interactions using surface fingerprints. And so that you are more aware what this problem stands for is, let's say that you define a target protein and you define the site that you want to target with your newly designed protein, and then you change the sequence of this of your candidate proteins so that they bind in a predictive manner to the site you specified. Now, why is this so hard, right? So that's sort of like, this has been one of the challenges that in computational protein design has been so like, oh man, we just fail and fail and fail. And oftentimes when we succeed by using a lot of in vitro evolution and whatnot. So, it's always sort of like a very dirty thing in a way. But it's not just for some reason, right? The reason is that oftentimes, and I think, again, it's a problem of representation. The way that we represent proteins when we're designing them, it basically ignores a lot of the things that are happening in solution. So, when we model these proteins, we don't really have any solvent. We also completely ignore that they're actually dynamic molecules. And I think this is the kind of problem where any little difference, any methyl group that you have in the wrong positioning is the difference between a success and a failure. And so, and that's why it's hard. Okay, but I think I'm sort of like probably taking too much time. So let me tell you what we did, and maybe I'll just show them a few cases. Oh man, I can do another presentation. It's okay. No. So okay, well, let me just sort of like tell you how we actually implemented this so that you have an idea of what's actually happening in the computer. And so what you see here is the pipeline. And so we often start with a target protein. Something that it's important that we do is when we have a target protein, we actually run our first predictor on it to try to figure out which sites are more likely to engage with other proteins. So we don't go just at any site, because somehow we found, we developed this algorithm to identify which sites are more likely to be bound. And then we extract target surface fingerprints, and then we try to figure out which, we create fragment databases with many fragments. So here you're seeing sort of like 100 million fragments. So like large numbers of protein motifs that, because the algorithm is actually extremely fast doing this matching, we can actually do this, in very short time frames. And then once you find your best matches, what you take out of this stage of the protocol is basically a pose of a binding seed. So that's what we call this this fragments that were located there. Let's see exactly. So you see sort of like the binding seed over there, and then we present these binding seeds into we mount them almost into scaffolds so that we can then sort of like do screening at least at the medium throughput level. So some examples. We started going after the proteins that were more easily accessible for us, right? So this one was accessible during the last years to many of us, either we wanted or not. And essentially what we did here was to basically target a site which was not the same one that it was engaged by ACE2, which is the receptor, the cellular receptor of this viral protein, but actually in a different site, which is here on the side. And so, and again, we start predicting the potential binding motifs. We create around 63 designs, and you know, you're going to see this a lot. And we get a hit in a good day, because in a bad day, this would not be a hit, right? As you can see, because it's a very marginal, very residual signal, which you really can only believe if you actually then do a competition experiment with ACE2, and you know, your model predicted that if ACE2 is bound, this one cannot bind, and in fact, you start seeing, oh, the signal is gone. And so when the signal is gone, you're like, all right, well, maybe this is right. But these are very residual signals that we're collecting at the surface of yeast. So for those of you that are not familiar with this data, this is the designs being displayed at the surface of yeast, and we staining them with a target protein. Okay, and so we start with something that it's not super great, right? So what do we do next? Because obviously, when something binds so residually, we cannot even do biochemistry. We cannot measure anything because it's outside of the range. And so what you see here is basically a bunch of iterations. Let me see if there's more. No, there's no more. A bunch of iterations to basically take this scaffold from something that was barely measurable to something that then was 4.6 micromolar, to something that ultimately turned into an 80 nanomolar binder. We then also validated if it was functional, if it could neutralize virus, which kind of strains did it bind. But I won't tell you too much about this because this is just basically functional validation. And basically what took us from a design that we could barely detect to something that binds 80 nanomolar was around 6 to 8 mutations. That's basically that. And I'll tell you more about what we're learning from these mutagenesis experiments. And I'll use another case for this. So now we're going to go to PDL1, which the interesting aspect of it for us is that basically it is one of the flattest sites that you can actually find in the PDB. So it's a very flat site. So it's not very easy to target this, particularly with small molecules, although antibodies, for instance, can do it very well. We again run our search for binding motifs. And we cluster these binding motifs. We can see that there are different levels or different types of interactions that can be that can be predictive or at least proposed as potential modes of interactions. We select one of them, and we start seeing that some interaction motifs start arising from this particular cluster, and we go again. And the story is kind of the same, right? In a good day, you have a match, or you have a hit. In a bad day, this would just be noise. But of course, that here, basically what we did is if you go and you do a point mutant in a predictive interface, does that binding goes away? And you see that it actually goes away. You see the same story in a different binder. And then again, let's maybe take a look at what we did here to try to learn something about these designs. And so what we did was basically what is called the site saturation mutagenesis, where we have the design and the interface that we engineered in. And so what we learned here was basically that in the binding motif that we had brought from the massive search, the side chains were close to optimal. So that's the data that you see here in these boxed positions. There aren't many other mutations which can actually improve your binding. Now, what we also saw is that in the rim region, and basically by this I mean that the mutations that are around this region and in other positions, that there could be a lot of other mutations that could be beneficial for binding. And so just to tell you how this actually went, so again, you see that there's a number of combinatorial libraries, some of them to optimize stability because this protein was terrible, some of them to optimize binding. But ultimately, a long story short, is that you start with something when you can measure it, which is around 2 micromolar, and you can push it up to 265 nanomolar. Now, I haven't shown you any structure yet. So let me show you some structures, right? So clearly here, we crystallized also together with the help of collaborators some of these designs. And what we saw was that, well, in fact, the predicted binding modes were correct with root mean square deviations of around 1 angstroms, and all the side chains that we thought that were involved, and we saw this for 2 different cases. And perhaps what, and so this was obviously a very interesting result for us. But what we also learned from here was from the comparison of the binding motifs that we were using here with the native binding motifs, right? Because one of the conversations that one can have, well, when you're doing these searches, are you just recovering the native binding motif that is used by the natural pair, which in this case is PD1, or are you actually creating a de novo interaction in the sense, in the structural sense? And I think this slide actually shows it very well, that you see that the native protein uses a beta hairpin while we were using alpha helical motif in order to engage this site. Okay, so in the remaining few minutes that I have, I will try to address a few shortcomings and how we actually try to improve the pipeline so that we could actually get more binders without having to do in vitro evolution and all these kinds of things. So one of the things that we noticed, and this is a common problem known in design pipelines, is that it's actually very challenging to engineer hydrogen bonds in your interface. And our site saturation mutagenesis results told us the same. So in the pipeline, we put a few more steps of enrichment for hydrogen bonds. And so, and what we saw now in this case is that in fact, we could actually take out of 1,000 designs, that's basically how many we tried, we could actually take 2 very clean binders straight out of yeast. Yet whenever we, and again, so this is the clean binder, the message from the site saturation mutagenesis is kind of the same, right? In your seed, in the hotspots, if you will, from the seed that it was grafted, you don't have a lot of space to negotiate the mutations, so the mutations are pretty much optimal. But in the edges, you still have a lot of molecules, or in the polar rim, you still have a lot of amino acids which are not optimal. And so essentially, we measured biochemically the affinity of these binders, and basically we got a binder which was about 6.5 micromolar, 2 micromolar, something like that. So again, it's not something to write home about because it's not a nanomolar or a picomolar binder, but it was obtained straight up from the computer. And of course, we should sort of like also keep it in mind that the native interaction between PD1 and PDL1 is actually 8 micromolar. So I will spare you a bit the details of the experimental characterization, and we'll close with this type of idea. So one of the questions that we have a lot, and I don't know if you're asking yourself the same thing, is, well, but this just works for helices, right? This helix thing, this has been totally solved 10 times. But the reality of the fact is that when you use the surfaces, they don't really care if it's helices or strands or loops. It's just a representation. They don't care what's underneath. So what we actually try to do is like, well, can we now expand to other motifs? And in the end, at the beginning, the reason why we did it with helices was just because we knew that down the line, to transplant helical motifs to helical proteins, this would be easier. And that's what we wanted to, in a way, facilitate. But the question still remained, well, but you guys cannot really do it with another thing besides helices. So we went ahead and actually tried to create different types of motifs to serve as binding seats for this design interaction. And here you start seeing that we start obtaining a different type of structural complexity in terms of having helical hairpins, sort of like almost like sheets, and creating, again, very large very large libraries of motifs themselves, right? And so again, what we saw here is that in this case, we could try to, or we did try to design motifs, and what we see here now is one of these de novo designs where there's basically no helical element into it, but rather this protein-protein interaction is completely based on the strand surface. And again, for all that's worth, whenever we actually try to predict the complex with AlphaFold 2, it does tell you that is in agreement. Which, if you think about it, it's pretty remarkable result for AlphaFold, I would say, assuming that it's right, because it could be wrong because we don't have a crystal structure. But this is a protein-protein interaction which you have zero co-evolutionary constraints. These two proteins are. They were just never together, right? You know, in evolution, you cannot take that signal from the sequence alignment. So, you know, the fact that these things actually agree with each other is kind of like, you know, it's almost shocking in a way. And again, what you see here is that we can have binding signals and very quickly just improve them to become even more expressive in terms of binding. When we do the competition to make sure that it's binding to the right site, it is competing with a reagent where we know that the site is binding, and then we have an apparent affinity, which is at the very acceptable level. But again, you shouldn't take these numbers as real affinities because they're not real affinities. This is just apparent affinities. Okay, and so I think now I seem to be almost on time, and all that there is in between food and beers is me. So I'm going to just close here, and I hope that I have explained to you and told you about our efforts to represent proteins in different ways and how we're using surface representations to try to engineer or design molecular recognition events which are very traditionally hard to design using the typical techniques that we've been using in protein engineering. Now, there's still a lot of problems, you know, as everything that we do in the lab when we design new methodologies, everything is super clunky and super amateur, but you know, it's not really, you know, we're not really computer programmers. We just really want to solve a problem. So clearly there's a lot of things to improve in this pipeline in order to make it much smoother. And again, you know, there's also interesting perspectives on how we can actually use these fingerprints for drug design. So with this, I will skip 100 slides and the most important one. So I have really been super lucky in having had the chance to work in Switzerland, a very interesting country to be a scientist, and also to have shared my times over there with students that not only were passionate and very committed to what they were doing, but also that pushed me to try to become a better scientist, which I think is always very important. And also, you know, having had the chance to have our research supported by a number of funding entities and collaborators that made this work possible. So I thank your attention, and if you have any questions, I'll be happy to answer any questions you have.
Awesome. The fingerprint stuff for the figuring out what area of the protein to target. How is it just finding hydrophobic patches? Like, what is it more subtle than that? How, like, have you ever have you ever discovered some kind of a surface hash that from the algorithm where it wasn't already known that a protein binds to it, and then you design a protein to bind to it, and I'm really curious about that.
Yes, absolutely. So,
Bruno, if you wouldn't mind repeating the question into the microphone so the folks online can hear.
So the question is related to when we have this first layer of prediction of the sites that are more likely to be targeted by other proteins. Have we actually found sites which we didn't know the ligands for them or the protein binders for them? So the example that I showed you for the RBD in SARS-CoV was one of those examples. Lo and behold, a few months later, some antibody came that binds to exactly the same patch. When we did it, nothing was there, but then some antibodies, and of course, antibodies are a little bit more flexible. They can do anything and everything. but it did happen. So, and then going to your first point, is it just hydrophobic? So a large fraction of the time, it is sort of like you do recognize a bit the hydrophobic patch, right? It's like, this is not the real. So in a way, sort of like you can kind of think of it as proteins are almost modular. Some parts of the surface are there for solubility, some others are there for the molecular recognitions and signaling and function. So oftentimes you have that. Sometimes it's less common, it's less obvious. So that also happens. So I think there is a distribution, but I would say that perhaps at least 50% of the time, like, yeah, this is hydrophobic. I get it. There's also this aspect that sometimes it's not just about the chemistry, but also the shape. So there's something about the shape that you're trying to target. And I think the algorithm picks up that a bit because, I've seen it, right? So it's able to pick up that a bit better than the human.
I have a related question to that. I know that it's a breathing surface, but it's sort of a different flavor of binding, where you want the binding to like do something instead of just kind of like stick and be there. So do you think that you're searching for like dynamics as part of your patch or not?
No, I think that it's very sadly for my. Oh, yeah. So the conversation is, can we actually find some sites that have dynamics associated with it and some type of allosteric regulation? I didn't really summarize very well, but to give just the key points, because I really need to go into this. This is probably one of the biggest frustrations. I don't know, at least it's mine. I don't know about the other protein designers in the crowd, but the fact that you don't know what your proteins look like, right? You have no idea what the dynamics of the molecule that you're modeling looks like, or at least in most cases you don't. And so in this case, I don't think our algorithm has any chance because it has never seen any of those, right? It was never trained, so it can only really learn things for which it was trained for. And in this case, it never really seen anything related to dynamics or to cryptic sites or anything like that.
Right. I guess my question is if you think that there's something intrinsic to the sequence or the shape that like makes a dynamic site look different than a non-dynamic site, even if you don't actually have that dynamic information. Do you think that's something your model's finding in like a latent space somewhere?
So I will give you, so yeah, so again, to clarify the question is, would there be any type of fingerprint that would, even if the site, even if you're not considering dynamics, that it knows about the dynamics of the site, and I will go back again to say with my very pessimistic me, no, I think it has no chance because it never really seen any kind of molecular ensembles, which is sort of like, it has no idea. That's my perspective.
Question about types of
Good question. Yeah, no, we have not tried. Right now, pretty much our pipelines are based on atoms that are most presently in proteins because there's more there. I don't think it's absolutely impossible because I think it's definitely feasible to encode the charges and the geometry of the DNA molecules. Of course, we have less data, which is always a little bit of an issue. So I think it's possible. We're never done. It's good.
One thing that I see for the first example of the block of the advanced because of the type of the relation patients and including the human. So I can think to step out a little bit in this example. Usually, that part is demonstrated. So that was the first thing that was a little bit surprising. And so the second question is the limitation that you see that makes the stability and the we have the area of the payment that actually stabilize the rotation that you're finding the. So second show intention setting up the final confirmation.
Those are all very important and relevant issues. So I should just tell that here, while we were trying to do exactly not having to deal with the scaffold design problem, we just really wanted to take things that were there, folded, looking beautiful for us, and just change their surface to enable some type of molecular interaction. As it turns out, this match, when we actually looked at it, it had the cavity of this side in the middle because it was actually a small molecule receptor. So essentially, when we started looking at well, and we started expressing the protein, it's like, well, we have an issue here because if the small entity was a small, you should see that cavity was like it was a nightmare. So like, all right, well, let's see what this what we can do with this and try to overcome this mutation so that we don't have to be working with this scaffold, which is so poorly behaved. And then I want to absolutely agree with you by saying that when we're stabilizing the protein, it is hard to imagine that you didn't stabilize or fit in the scaffold itself, and that didn't have an influence in binding, right? The only aspect how I can counter that is by saying, well, but look, whenever we were going to do the point mutants in the surface, we could still eliminate. But I think clearly there we gain, and I've done a lot of work in stabilizing motifs, and I know that if you stabilize a motif in the bond configuration, you're going to get logs in it. It's not a joke. Right. And I think here it's probably part of it. It was also.
You probably could have seen it if you were looking at entropy combining with ITC or something. Would you have actually decreased entropy of binding?
Well, I think we did, but not explicitly, right? I think we by stiffening the scaffold and by stabilizing the scaffold, we likely did. I would be shocked if we didn't. But it wasn't really explicitly for that. But I think this is a very important point, and it's actually part of the picture, which we are completely missing from our design process.
Bruno, really, really exciting stuff. When you're screening and designing these larger proteins for binders, are you normally doing the first pass by like a yeast display with clones, I'm guessing?
Yes.
For some of these helical proteins that are really stable, do you ever see like that they don't display very well?
I should have brought in slides. This was the surprise of my life. Because you always sort of like assume that, oh yeah, they're like so great proteins and whatever, and it's going to be great. So we did this experiment in so now basically what I told you is that every now for each target we basically do 1,000 or 2,000 designs, right? We encode them with these oligos, we screen them, and the other day one of my students, Anthony, in fact, showed me this result. He said, I wanted to check how many proteins are actually displayed, and it was less than 50% of the designs. Which, of course, it could also happen that we're crappy designers, right? We just design, I always, when things don't work, I always design, I always like, blame the designer, right? It could be that we just put too much hydrophobics on the surface or whatever it was, right? So that's not impossible. But it is also not that you get to see all your designs making it to the surface. So I guess you're asking us, the question is because you knew that the answer was going to be this.
So maybe the leading answer is, we run into a lot where hyperthermal stable helical proteins, especially when they start to get large, display like garbage. And actually a lack of display might mean that the design is working really well. And sometimes the ones that come through and display are the ones that actually didn't work that well. So you actually might be the best stuff actually you might not be seeing because you're doing yeast as the first pass.
Now of course, yes, maybe, and I would love that would be true, but but let's imagine that it's just sort of like a scaling factor, and that in fact you're just missing twofold because you displayed half of the design. So rather than having 3 designs that succeeded, you had 6. Well, I would be happier with 6, but in terms of the jump, it would not be super huge. But again, it could also not be sort of like a clear-cut distribution, but it is true that we observe this true as well. And our assumption was that, well, look, these proteins are stable, they're soluble, well, we designed them, part of them, so we thought that, you know, Chris did a good job, so. And so, you know, it was a little bit of a surprise, but I guess, you know, perhaps we're starting to connect more with this, just saying like, you know, it's not a surprise, it's maybe what you would expect from. Please go ahead.
Binding to other sites that you haven't designed them to bind to.
We only had one case where we saw like a binding event that we couldn't compute. So it's not sort of like, it didn't seem like generally when we design stuff, you know, we don't have a lot of, you know, we don't have like huge success rate, like, you know, right now it's still sort of like, no, we wish that things would work a bit better, or a lot better actually. But when they actually work, they seem to work by the right reasons. You know, and I think in a way, right, you know, a lot of people actually say like, wow, how do you actually design proteins and whatever? Well, one of the reasons why we design proteins is because we're designing things for them to work for the right reason, rather than just sort of like attaching to running bounds. Because it's clear thing, engineers and running to attach to the other ones, right? So oftentimes when we see things working, it seems, at least from the experimental evidence that we gathered, that it's for the right. No. You know, and I think, you know, this is, I think, so I know that, you know, you're just starting, you're also, and, you know, the world has a lot of possibilities, and the world does have a lot of possibilities, and we should never give up trying new things. But this has been one of the promises in the field, where we're going to site saturate everything, and that's going to enlighten. Well, that didn't work. Until now, yes. But I do think that there's something there, right? There is something there. And I think, you know, part of the reason why it might not work out is again related because whenever you do this mutation, there's a lot of things that happen to this molecule which are not modeling, right? So it's actually unfair to ask your model to predict things that you're not modeling, right? So, and then, you know, I also just wanted to sort of like cautiously towards something which I think is kind of important, but, and I think other people perhaps are better than me to talk about these things, but I do think that the display level influence immensely the binding signal that you get, but like, you know, in a way that it's almost like, whoa, this bonds so well, and then measure the thing, it's like a micromolar bond. So you see that I think there's a number important for these kinds of exercises is to figure out what is the limitation of your screening platform. So I think it's something that it's there to be grabbed. So somebody needs to do it. Yeah. I think so, like the levels of display, you know, even any, perhaps at least this, if you're in, you know, I guess when the page is a little bit more limited, like, you know, the number of copies vary, but it doesn't vary widely. But I think in least, I think the solidity effect is absolutely. Again, I'm not the world's expert, and I'm not. This is saying this. I would say that I would not.
People say it's 10 to the 5. If you actually do a computation of what's the surface area like of a protein on the surface of yeast, if it's 10 to the 5 copies displayed, it's one protein every 10 square nanometers, which is like this it's pretty spread out, even if you have 10 to the 5 copies.
Yeah, I see. So basically what you're saying is that, look, but you know, but I think I'll just give you a more example of that is not really running that of the calculation, but I have to speak that way, but you know, for instance, even when you use a fab and then you use it 92, just to select It's something, right? So the fact that is, normal guidelines, which is an emergency, that is valid protection. The difference in signals is huge. Right, so, I see about the window, but just even with this little trick of either a valve or an IGT, you see this much of a difference. It means that they have to look closer than what you're saying. There is some kind of ability. Chris is, yes.
Have you actually measured the concentration of?
My conversations with Daniel, sure.
Sure, okay,
I-
well, if
I-
anybody would have done it,
Yeah.
we would have done it.
I can also tell you that, I mean, some of our affinity maturation campaigns by AirProne, we've seen synonymous mutations improve affinity, apparent affinity on yeast. Yeah, yeah, exactly. Yeah, apparent affinity on the surface of yeast has been improved by synonymous mutations, which presumably are deflecting and only impacting display percentage.
Yeah. But now that perhaps could be normalized by the display level, right? I guess if you get like really good, and if you have a display tag, and if you have a good way to then sort of like normalize this, maybe then at least you could remove some of that. So at least one will go.
What was your ligand, a huge ligand or something?
We can talk afterwards.
Yeah.
In general, and that's the we have not tried. I think, now, of course, seeing the results in DNA, I think everybody sort of like thinks, and I think we also think that that's probably one way to improve solubility and stability of protein. And that's the right thing. So we are sort of thinking about how to improve the way it is. But now, Chris says, oh, it's very too stable. That's also not good for yeast. So we have a little bit of a conundrum here. Maybe we don't want to do very too stable. But I guess we'll see. We'll find out if this actually will increase the display levels.
Post translation. So the field of protein is too stable. That's leading to conflicts. Sometimes you see that maybe maturation is breaking the protein.
Well, that's great. I see, and it's funny because also these proteins are pretty small, right? But apparently they really need to be unfolded to go through the display machine or something. I see.
I was curious with the massive experiments. I guess I'm curious about that. How much could you get from you showed exactly what you want to break down. If you don't break it, it's not going to take it's not going to be
Well, so I tell you one thing, when you have very concave shapes, how can I tell you? So I'm just going to be sort of like thinking while I'm speaking, okay? But when you have very concave shapes, oftentimes what you run into problems is to find stuff that can fit it. You can actually fit a lot of motifs there, but then if you have a sort of larger protein, like the three-dimensional constrictions that they have, they cannot really fit it 90%. So I don't, you do have a few sites that are a bit more controlled, and like we seem to be able to also target it. Sometimes not so much also with clinical motifs, but actually with data science. So I don't think it's no, it doesn't seem like, in general, it doesn't seem like, I think what would be fun is actually to be targeted at the combat side. That would be fun, right? Which clearly, so like, an antibody is like, normally designed to do that, but I think, that a convex site to me kind of like, oh yeah, that sounds like a challenge. We've never done it. Yeah, that's, I would say, double like if I could put it, because, work with, I think that's most of what it does. Is that it does not unfold. But I think it does a little bit sort of like, the glorified modern poles, which are modern poles with more about the things, because they're not only understood. That's what it is. I think that's what, if you want to sort of like summarize the one thing, that's what it's.
Let's thank Bruno.
Oh, we need to treat everybody the same. I know, it's good. Well, we'll take one more. Is that okay?
Yeah. Let's
All right.
I was wondering if we could method to describe what people often in the steps, the break up design. That think that is stable for people online.
So, clearly. This was, the subject about the first situation where he was trying to figure out how to navigate the things. But right now, what we see that clearly our rendering set we don't understand engineering at the final. I think that's the way because I just described, partly we're not doing so many problems in this, but the other side of the staff itself. But it does, at least in the platform that we're using, we do have to. And it's coming out and go back. But I feel like the biggest bottleneck is trying to design and be reliable. And I always like to summarize this problem. This is very simple thing. If you have a message in the outside of it, right, that's enough for you to see a signal or not. And then it's just enough for the signal.
Okay, let's thank Bruno.
Thanks a lot. Thanks, guys.
I have one announcement about our next month speakers. We have a trifecta of speakers next month on March 8th. We're going to meet over at the HMS campus where we were last month. 3 people from MIT. We're going to have Gabriele Corso, Anna Stark, and Bowen Jing are going to talk about their title is DiffDock, Diffusion Steps, Twists, and Turns for Molecular Docking and Beyond. I'm really excited about the beyond part. So yeah, tune in next month to join us then. For now, that's what we're doing on my class.
Thanks a lot. Where are we?